My name is Zachary, and this summer I focused on alleviating issues associated with researching diatoms, a type of aquatic algae. And we applied a software called Visual Spreadsheets, and it's designed to conduct automated particle analysis. Diatom research is a highly time and expertise-intensive procedure. However, it's important because diatoms serve as a proxy for environmental health and change of aquatic ecosystems. This study is one of the first to directly apply Visual Spreadsheet automated particle analysis software to high-resolution microscope images, thus making it as a proof-of-concept study. And the preliminary work of this project will hopefully aid scientists and academic institutions and federal agencies in paving the path for automated species identification of diatoms. As I mentioned, diatoms are aquatic algae, and they are found in fresh and marine waters. They're microscopic, ranging from 2 to 500 microns. And one unique characteristic that they possess is that due to the hydrated silicon dioxide of their cells, they have a transparent property. Diatoms, they are abundant throughout Earth's marine and freshwater ecosystems. And as primary producers, they are also responsible for about 20% of the Earth's global primary net productivity. In addition to that, they're also responsible for about 20% of the carbon fixation throughout Earth. For our interest, we're more interested in their role as serving as proxies for environmental change and health of marine and freshwater ecosystems. So I want you to look at these two sets of diatoms and guess which pair has the same species. As you just saw, diatoms are very tricky organisms to classify. There are over 200,000 identified species, and diatom scientists, they have to look through these images on a daily basis at hours at a time. And this is very tiresome, thus motivating a need for an automated process. The main technique that scientists use to analyze diatom samples is through traditional light microscopy. And through this process, scientists are able to obtain high-resolution images by being able to adjust settings in the microscope, such as contrast and sharpness. So they're able to get very in-depth, close views of the cell structures and analyze them more in depth. However, this technique is very time-consuming and requires a lot of expertise from a limited number of scientists. So this also motivates the need for an automated process. For this study, we focused on Visual Spreadsheet software, and it is a software designed to conduct particle analysis analysis. And this software is typically used in conjunction with its complementary hardware, FlowCam. However, this is the first time it's being used independently of the hardware, so this is adds to the fact that this is a proof of concept. It allows for setting configurations, and so one of the main objectives of this project was to see how different operating conditions affected the quality of the software's ability to crop and analyze images of diatoms. And some of the measurements that this software takes includes diameter, length, and transparency of cells. So one way to think about this project is to imagine facial recognition software that you might see from a crime TV show. So in facial recognition, the software will look for unique facial features of someone, make an analysis, and try to identify that person based upon those features, usually from a database containing faces. For this study, the software focused on different pixel color differences in the particles to determine if something in an image was either a particle or a non-particle. Future work of this project will involve developing a library or a database of diatoms and be able to conduct automated species identification. So to start off, microscope images of diatoms were captured by a traditional light microscope. Here's an example of just one of the images that were taken. After images are stored, we rename them so that the software could properly process them. And we wanted the software to follow it in a sequential order, so we changed the file name so that the prefix would follow with an image name followed by a different numerical value that represents its place in the sequential order. After that, setting configurations were made. And so the two main things that we focused on were distance to nearest neighbor. This setting basically defines the minimum distance that two pixels can be, the minimum pixel distance that two particles can be from one another before they're incorrectly classified as one particle. So it can, if the distance is too short, two diatoms could be counted as one, and that's not what we want. In addition to that, we looked at segmentation thresholding. So in image analysis of grayscale images, and in our case, are the microscope images, you have a color pixel range ranging from 0 to 255. A value 0 equals black, 255 equals white, and every value in between equals a different shade of gray. The software conducts a process known as thresholding, and this process is used to distinguish foreground or objects in the image from the background. So in the case of this software, if a diatom particle or pixel in the image express a pixel color value greater than the user-defined threshold value, the software saw that as a particle. If the pixel in the image was less than that threshold value, it did not get picked up by the software and was deemed as a non-particle. Following that, images were directly imported to the software simply by using the File Process button as indicated by the red arrow. Afterwards, the automated process of analyzing the images occurs, so the user has no control of this. Here, on the left picture, you'll see a blue box surrounding a diatom, and that shows what the software has identified as a particle and is attempting to take a crop. Afterwards, in the image next to it, you'll see it's been conveyed right here in the next window. Cropping particles is important because you only want one diatom in one picture, because the software will gain information from the pixels used. So you don't want multiple pixels from different particles in one image. Afterwards, statistical data are presented on the home screen, and this information might include the total particle count and the amount of time it took to sample. Here, the results of the analysis are also displayed, but this contains the cropped images, so the user is able to view and look through all the images that the software has attempted to crop out. And below it, on the left side, you'll see a pink coloration. That's something known as binary image overlay. And basically, that's what the software has deemed as particle, and that's the information that the software will see to make an analysis and measurements of the diatoms. It's important to note for future discussion that there's only the outline of the shapes that are pink. We want the entire particle to be pink, so both the outlines and the interior. So that's something to keep in mind as I continue on with this presentation. After that, this method was repeated multiple times using different conditions, because one of the goals of the study was to see how different operating conditions affected the study. And close to 250 trials were done with this, so very interesting. Afterwards, a visual assessment of the cropped particles, as indicated by the left screen, was taken to see how the software did with cropping. Here I have two graphs that focus mainly on the distance to nearest neighbor. So again, this is the minimum pixel distance that two particles can be from one another before they're deemed as one particle. And you'll see as the distance to nearest neighbor on the left graph increases, the time also increases. We don't want that because the goal of this project is to make diatom research more efficient and quicker. So we don't want that. In the graph to your right, you'll see that as the distance to nearest neighbor increases, the total diatom particle count decreases. Again, we're trying to make diatom research more efficient and quicker, so we don't want that. It is significant to point out, though, that when the distance to nearest neighbor is at a lower value, such as 1 or 2, the software yields much better results. For example, in 68 seconds, the software was able to identify 216 diatom particles, and in contrast, it took 400 seconds or 6 minutes and 40 seconds to find 255 particles. That's in about a 5-fold efficiency difference in an analysis. So here's an example of how the microscope images are analyzed, and you'll see here in the left image that there are multiple diatoms. After analysis, the software is able to identify multiple diatoms and crop them out. Visual assessment involved deeming if something was a correctly cropped particle or incorrectly cropped particle. Correctly cropped particles only had one diatom per image, and the more binary image overlay or pink you had, the more statistical data that the software could pick up from that particle. Incorrectly cropped particles either had multiple diatoms per image, so in the lower right corner you'll see multiple diatoms, or you'll have a singular diatom that was fragmented into multiple crops. You don't want multiple fragmented crops because that adds to the total diatom count, thus throwing off your results with inaccurate data. The software did a pretty good job with darker or heavily silicified diatoms because the pixel colors that the diatoms expressed had a pixel value that was greater than the threshold. Therefore, the software saw it easily as a particle. In contrast, the lightly silicified or lightly colored diatoms had pixel colors and images that were less than the threshold. Therefore, the software struggled to crop them or didn't pick them up at all. Finally, the last image to your right is an example of how two particles have been cropped as one individual diatom particle. And we don't want that because that throws off the total diatom count. One of the most important factors or aspects of this image analysis is the binary image. The binary image is something that results after the comparative analysis of the pixels occurs in thresholding. And so in the lower picture, you'll see a black and white image. Those white particles, the white coloration you see is what the software has deemed as a particle. And everything else that's black is something that is not read or processed as a particle by the software. This is an issue because, as I mentioned before, we want the entire color, the entire shape to be filled because that's how we get our statistical data for the software. And this is the result because diatoms, due to their transparent property, they display similar color values to the background of a microscope image. So this brings issues to what we need to work on for future work. Here I compare the difference between how other attempts at automated processing with Flowcam hardware is with our method of just directly applying the images. So you'll see that in the top image, that's an example of Flowcam. And basically that method uses a flowing medium of water that passes through a tube, intersects a camera in a microscope, and the images are taken. But due to the low resolving power of the Flowcam, the images are low resolution, which hinders the ability to efficiently analyze diatoms. In comparison, our work directly imports high resolution images for better analysis. In conclusion, the software is efficient in counting diatoms. So there was about a 5-fold efficiency difference between manual analysis and automated analysis. However, much improvement must be performed for the binary image overlay because this is the information that the software uses to correctly analyze the diatoms. So this future work might involve working directly with the program developers and preparing microscope slides so that diatoms aren't touching. So overall, this program could be used to complement traditional light microscopy, but should not be used independently until improvements are made in the software. I would like to thank the NSF, UNAFCO, and the NSTAR Diatom Lab and Fluid Imaging Technologies for their help and support. That's the end of my presentation. I'll take any questions.
We have folks on the WebEx.
I was just curious about a couple things. One was, it seems to be like all your slides have single diatoms, like they didn't happen to sit on top of each other.
Yeah.
Is that selectively done by how the slides are prepared? Or?
So that is one of the issues that the program can run into. So as I mentioned for future work, preparing the diatom valves so they're not touching is key. So if they're on top of each other, the software will only see the bigger outline of the bigger diatom. So those two will be incorrectly processed as one diatom particle. So that's one of the future works that we have to collaborate with the software developers.
And then the second question I had, because you were talking about the health, like the water system.
Mm-hmm.
Do these diatoms, they pass away, so to speak? So are you sometimes just looking at bodies that aren't necessarily viable anymore, or? Because I'm just curious if if your method can discriminate that way and say, oh, we're looking at dead diatoms versus live ones.
So these microscope images that we had, they're actually the remains of the diatoms. So all the organic matter has been removed, so they're technically dead. But that's some of the future work that we have to do, is to see if we can include live images or high resolution images of the live diatoms. So that's also some more future work that we plan on looking into.
And is there a way to optically discriminate when they're live versus dead right now that you know of, or not necessarily?
As of right now, I'm not sure, but as I said, we'll be working on this more during the next year or two, and hopefully that will yield some good results.
Great, thank you.